Our next presentation is Accelerated Biosciences, and I'd like to welcome Yuta Lee, founder and CEO, to the stage.
Hi, everybody. My name is Yuta Lee, founder and CEO of Accelerated Biosciences. Today, I'd like to spend a little time to tell you about the human trophoblast stem cell, or HTSC. This is the earliest cells that you could possibly source without any ethical issues. And we are providing it as a stem cell source for everybody here for their therapies. So we are a stem cell platform company based in Philadelphia, and we have full freedom to operate over these HTSCs. And my job really is just get them to GMP, which we completed back in April this year, and package it up with the CMC documentation and work with you guys on your to help you accelerate your programs to market. Now, as we all know, the biggest problems that we face today is finding the right stem cell source to start with. You face ethical issues, inconsistent cell source. You have to choose between scalability and immunogenicity. And many times the FTO or the IP is unclear and the CMC is kind of a mess, especially coming out of academia. We actually have solved all of these problems. And our cells have pretty interesting characteristics. They show markers all three germ layers. They're ethically sourced. They're scalable. They have natural immune privilege. They're pathogen-free. They have very stable genome to high population doublings, and we have a large IP estate to protect it. This here is a continuum of fetal development. Our cells, the HTSCs, are actually sourced from the embryonic stage, 4 to 8 weeks post-fertilization from an ectopic pregnancy. And as you guys already know, the earlier you get the cell, the more biologically active they tend to be. On the left, you'll see a blastocyst at 5 days, right? And our cells come from the trophectoderm lineage, the ring from the outside, which later becomes the placenta. And what happens in an ectopic pregnancy is that the embryo is traveling down a fallopian tube and it gets stuck. And it'll keep growing until it bursts the tube. And if it bursts the tube, the mother may die of internal bleeding. It's actually the highest cause of death in first trimester pregnancies, right? So what happens is the mother will feel sharp pain, go into the ER, get scanned, and doctor goes, oops, you actually have a tubal. We have to schedule an emergency salpingectomy, where they go in and ablate both sides of that mass and use a scope, pull it out, give it to the pathologist to check, yep, it's an ectopic, and they throw it away as medical waste. Why do they do that? Well, on the left side, the embryo, the blastocyst, actually needs to implant into the mother by day 7 or day 8. But you don't discover an ectopic until 4 to 8 weeks after. So there are many weeks in between where that embryo has zero connection to the mother, no nutrients coming to it, so all OBGYNs understand it to be a non-viable fetus. Okay? So what we do is, when we perform the salpingectomy and we pull out the mass, we actually just scrape off the chorionic villi, which is the preplacental tissue on the outside of the embryo, and we hand the mass back to the pathologist to check it, and they still treat it as medical waste. So that's why we have the earliest stem cells that you can find without any ethical issues, and we're ready to send that to you guys to work with. Okay. A couple of interesting characteristics. We can scale these cells out to 85 population doublings just using off-the-shelf media. Okay. And 85 population doublings is e to the 25, or if you use 100 million cells per dose, is e to the 17 doses. I mean, that's enough cells to create 2 trillion human beings. So we've solved the scale problem. Our cells also naturally express HLA-G because we get our cells from the preplacental source, right? HLA-G, as Nkilt was saying earlier, is one of the main markers that prevents the mother's immune system from rejecting the embryo, because the embryo is partially the father's genes and the mother's genes. So it's an alien thing, right, trying to attach itself. Well, the HLA-G dampens down all of the mother's immune system and allows it to stay attached and grow into a baby, okay? Now, when we differentiate our cells to neuroprogenitor or pancreatic progenitor, the HLA-G sticks, which is amazing. So we've done 5 different animal studies, all with human cells in immune-competent rats. Not only do they survive, they home and graft, and we see no teratoma or tumor formation. We get all the behavioral effects. These cells are super stable genetically. At 72 population doubling, which is the last time we tested a karyostat, completely stable. And we believe the reason is because these cells have been incubated in a fallopian tube. It's never gone into the endometrium, to be affected epigenetically by the process of term, right, or gone through becoming a human being. So early naive cells that have been preserved in the fallopian tube. We currently source 15 GMP-compliant cell sources or donor cells from Taiwan. And in Taiwan, a third of the women that come in have HBV, so hepatitis B, and more than 90% have CMV. Okay? Now, when we draw the blood of the mother and we test for HBV, we have to throw the cells away. This is according to the FDA rules. Now, what I did was I took all of those samples and I sent it to IDEXX for the H-Impact-1 to test for 25 human pathogens, and they come back completely negative, which is great. That means that there's a defensive mechanism at the fetal-maternal interface that we don't fully understand yet because we can't do experiments on healthy pregnancies, right? But also means that our cells as a starting cell source is probably the cleanest sources you'll ever find. When the mother has HBV or CMV, the cells do not. Okay. We've also used an mRNA technique to turn our HTSCs into an iPSC, and then we took a standard kit and drove them to an NK cell, and they are naturally activated, and they kill like gangbusters, which is great. So if you're looking for an iPSC, consider this as a platform to start with. We already have over 100 patents, 54 granted already. Yeah? So a lot of protection for this, all working with Wilson Sonsini, one of the larger firms in biotech. We have 3 programs running. One is preclinical Parkinson's disease. And our advantage is that we are able to take our HTSCs and use a 1-day protocol to turn them into neuroprogenitors with TH-positive reading for dopamine. Our second program is type 1 diabetes. We're doing this in Singapore. And our cells also have the natural ability of producing insulin when you do a glucose response test. Now, people think, well, that's kind of strange. Why does it do that, right? Well, if you think about it, it makes perfect sense. When an embryo is attached to the mother, what happens when she takes a big bottle of Coca-Cola? How does the embryo then deal with the hyper and hypoglycemia resulting from it, right? Well, it turns out the placenta has the role to regulate everything coming into the embryo so that it won't get ejected or it won't die, right? And so when you do a glucose response test on our cells, insulin naturally jumps up, which means that compared to other programs, we can save the entire process of differentiation, all the rigor to get it to a beta islet cell. And the third program, we're working with the National Institute on Aging, which is part of the NIH, on cell senescence. So as the last part of my talk, I want to direct your attention to our work with them, and I'll show you some data that we produced. Okay. So the NIA is focused on solving cell senescence. This is pretty much, as you get older, there are a lot of stubborn cells that refuse to die, and they keep accumulating in your body, okay? And they keep putting out cytokines that they call SASPs, or senescence-associated secretory phenotypes, SASPs, that are affecting your body in very negative ways. And they're connected to Alzheimer's, Parkinson's, diabetes, a lot of different chronic diseases as well. Okay? So what we want to do is either downregulate them or get rid of them. Okay, so the whole thing started from this idea of heterochronic parabiosis. Now, I'm sure many of you have heard this before. 20 years ago at Stanford, they did this experiment where they took 2 mouse, and they glued them together, or they stitched them together so it would have one circulatory blood system. except it's one old mouse and one young mouse, right? And it turns out that the old mouse was getting biologically younger, and the young mouse was getting biologically older, which is so strange, right? They did another experiment where they only exchanged the plasma, and the same thing was happening. What does that mean? It means the rejuvenation of the old mouse is purely a function of the proteins in the plasma of the young mouse, okay? So because of this, the NIA researcher came to me and said, Yuta, I hear you have the earliest cells that you can find without any ethical issues. I said, yes, we do. Well, I'd like to know what would happen then if you applied the secretome of the earliest embryonic state stem cells on cell senescence. Because if young proteins from a young plasma can rejuvenate old mice or old mouse, maybe these things will have an even more dramatic effect. And that's what we did. So I'll quickly go through the data. We did 2 pilot studies where we had used WI38 cells and radiated them to mimic aging. And we put culture media on it. And in the second one, we isolated our EVs, or extracellular vesicles, and put it on the cells. And turns out we can downregulate all the mRNA activity. So because of the pilot studies, we went into a deeper study using 3 steps, characterize them in vitro and in vivo. And here is the transcriptomics. Turns out that we can downregulate all the SASP mRNAs, and this is volcano plot. You see all the green on the left side. And also, using Olink, we looked into the proteomics, and we were doing the same thing. All the SASP proteins were all downregulated. Okay. Then we looked into NF-kappa B, which is one of the activators of SASP, and turns out we were downregulating that activity as well as providing defensive mechanisms for double-stranded DNA breaks. Then we did a small N equals 20 animal study, and we got the same results. And so what we did was we kind of narrowed it down to 4 key markers, GDF15 for aging, CXCL1, DPP4, and BAFF, and these are pro-inflammatory markers that you normally see in aging. And we did a search, and they're related to all of these diseases, okay? And this is just using the exosomes. So we are going after hypertension as well as psoriasis for topical use, but there are all these other indications available. So if you are interested in starting a program using exosomes for this, come talk to me. So we have 3 platforms on offer. Our original HTSCs, which were already manufactured the GMP. So we're providing RUO cells, or research use only cells, from the same donor line. That means that if you take the RUO and you succeed with them, you just have to come back and get the GMP, and you're on your way towards an IND. Then we have our iPSC, as well as the exosomes coming off the original HTSC, all of them from the same GMP donor line. So we have perfect product market fit. We have early embryonic stage cells without any ethical issues. We have a platform with scale and immune privilege. We've de-risked the FTO with robust IP. And we have IND-ready CMC documentation ready for you, okay? And we just want to help accelerate everybody's product time to market and really just make CGTs, or cell and gene therapies, affordable for everybody, yeah? So if you're interested, you can scan the WhatsApp or LinkedIn, and I will send you the slides, and come talk to me. Thank you very much.